In this presentation, we'll learn about how plants convert light energy to chemical energy in photosynthesis. Remember back to our discussion in a previous lecture about the first law of thermodynamics? We noted that energy can be neither created nor destroyed, but can be converted from one form to another. For the vast majority of organisms on Earth, sunlight is the ultimate source of energy. Changing this energy from light energy into chemical energy is a complex process, but one that plants have excelled at. Organisms that use sunlight to make their own chemical food are called autotrophs, and autotrophs are typically eaten by heterotrophs, or organisms which must obtain carbon compounds from consumption of other organisms. Let's start by looking at how the morphology of plants enables photosynthesis to happen. In plants, photosynthesis happens in chloroplasts. Many different types of cells have chloroplasts and photosynthesize, but most photosynthesis happens in the mesophyll cells within the leaf. We'll look at different anatomical features of leaves later, but for now, notice that leaves have air spaces within them that are connected to the environment through small pores called stomata. Carbon dioxide, oxygen, and water vapor can pass through the stomata in order to facilitate photosynthesis in the mesophyll cells. A photosynthesizing cell typically has many dozens of chloroplasts spread through the cytoplasm. Like with mitochondria, chloroplasts have two membranes, an inner membrane and an outer membrane, and a space between them called the intermembrane space. Within the chloroplast, the liquid portion is called the stroma, and there also exists stacks of thylakoids called grana, or grana for singular. The thylakoids are a network of membranes connected to each other with the thylakoid space within. As we learned with the mitochondria, the membranes themselves are vitally important to support the enzymes and chemical gradients necessary for photosynthesis. Now, photosynthesis is a very complicated set of reactions, and so I wanted to start by giving you an overview of photosynthesis to help you keep everything in context when we dive further into detail. We can think of photosynthesis as happening through two sets of reactions. The first set are referred to as light reactions, and they occur in the thylakoid membrane. In these reactions, light is used to excite electrons in chlorophyll and send these electrons through an electron transport chain. Like with respiration, these transport chains pump protons in order to generate ATP and an electron carrier called NADPH. These two high-energy molecules are then used in a set of reactions called the Calvin cycle, which bonds together carbon dioxide molecules to produce sugar. Remember that breaking up sugar molecules in cellular respiration is an exergonic reaction that releases energy. In photosynthesis, the assembly of sugars is the opposite, an endergonic reaction that requires the input of energy. This energy comes from the high energy molecules produced by the light reactions. The assembly of sugar molecules from carbon dioxide molecules is referred to as fixing carbon, or carbon fixation. You can see the overall equation for photosynthesis here. You can see that carbon dioxide and water are inputs, and sugars, oxygen, and water are produced. One little side note is that water appears on both sides of this equation. This indicates that the same molecules of water that are used as inputs are not used as outputs. The output water molecules are different and newly formed during photosynthesis. They form as a byproduct of reactive oxygen species, or ROS. If we simplify the equation by taking out the water from both sides, we can easily see how the equation looks in terms of the redox equations we saw before for respiration. The carbon dioxide is the oxidized form of carbon, and during the reaction it becomes reduced to sugar. The water is the reduced form and gets oxidized to oxygen molecules. Visible light is part of the electromagnetic spectrum. In fact, it's just a very small part and can itself be divided up by color. Different colors represent different wavelengths of light, with longer wavelengths tending toward the red and shorter wavelengths toward the blues and purples. When light hits an object such as a leaf, some of that light is absorbed by pigments and some of it is transmitted or reflected. The wavelengths of light that are transmitted from the object combine to form the colors that we see and perceive that object to be. When white light strikes a green leaf, some of the wavelengths are absorbed by the leaf, and the green wavelengths, or a combination of wavelengths that produces a green color, are transmitted to our eyes. And this is why we see leaves as green. Leaves actually have several different pigments. The main ones are chlorophyll A and B, which are almost identical in structure and appear as green because they don't absorb green light. Plants also carry carotenoids, of which beta-carotene is one. These are very different and usually oranges and reds. Tree leaves appear green because most of the pigment present is chlorophyll. However, at the end of the summer, when a tree stops producing chlorophyll, the chlorophyll degrades and we start to be able to see the carotenoids, which is what gives trees their fall colors. When a photon of light strikes a pigment molecule, electrons are excited and elevated to another electron shell. The amount of energy required to excite an electron in this way depends on the pigment itself and how excited that electron gets. The lower energy red wavelengths are able to excite an electron into a high energy state, but blue light, which carries more energy, excites electrons into even higher states. Furthermore, different wavelengths are absorbed by different pigments. The different wavelengths that are absorbed are called the absorption spectrum, and this basically tells us which frequencies are absorbed and which are transmitted by a given pigment. A related concept is the action spectrum. The action spectrum indicates which frequencies are actually absorbed to carry out photosynthesis. You'll notice a lot of similarities between the absorption and action spectra. Both spectra show a lot of absorbance and activity in the blue range and the red range, but relatively little absorption and little photosynthetic activity in the green range. It makes sense that these spectra are similar because photons need to be absorbed in order to be used by photosynthesis. Notice, however, that the action spectrum is broader than the absorption spectrum for a particular pigment. Plants make use of all of the pigments in order to photosynthesize. Chlorophyll A and chlorophyll B are very complementary to one another and are both directly used for photosynthesis. Carotenoids, however, may also play a role in photosynthetic ability, but their main function seems to be in preventing cellular damage caused by light. When light hits chlorophyll, electrons may be ejected, causing free radicals that are able to react with and damage the chlorophyll. Carotenoids seem to be able to neutralize these free radicals before they're able to do any damage to the chlorophyll. So let's look at what happens when a photon excites an electron and moves it up to an excited state in a pigment molecule. The first possible outcome of this is that the electron simply falls back down to its ground state. When it does this, it may release heat or it may release light, possibly at a different wavelength. This is what we see as fluorescence. Another possible outcome is that the energy is transferred to nearby molecules through a process known as resonance energy transfer. In fact, chlorophyll molecules are arranged specifically to facilitate resonance energy transfer among them. The energy may be transferred around from molecule to molecule until it reaches the reaction center at the middle of the so-called antenna or light harvesting complex. The antenna complex is arranged like this to maximize the light gathering ability and eventually transfer the resonance energy to the reaction center. When that energy reaches the reaction center, or when light hits the reaction center directly, a redox reaction happens as the electron is transferred from the chlorophyll to another molecule that we call the electron acceptor. Here's another view of the light harvesting complex as part of a photosystem. You can see that when the photon hits the chlorophyll molecule, their energy is passed through the light harvesting complex to the reaction center. When molecules at the reaction center are energized, the electron is ejected from the chlorophyll and is rapidly taken up by a primary electron acceptor. So let's see what happens as the electron is ejected from the chlorophyll when a photon hits. Once the electron is excited in the chlorophyll, it's very quickly taken up by the primary electron acceptor. Now that the chlorophyll molecules at the reaction center are positively charged, they very quickly oxidize water in order to replace the electron in the chlorophyll. When they oxidize the water, oxygen gas is released. This is what plants breathe out. Once the primary electron acceptor has the electron, it passes the electron to plastoquinone, or PQ. The electron has been delivered to an electron transport chain, which generates ATP in exactly the same way we saw with cellular respiration. In other words, the transfer of electrons down the electron transport chain is used to pump protons into the thylakoid space. The buildup of photons in this thylakoid space causes a strong electrochemical gradient to build up between the thylakoid space and the stroma of the chloroplast. This electrochemical gradient is used to generate ATP through ATP synthase. This is just like what happens during cellular respiration. When the electron reaches the end of the electron transport chain, it goes through plastocyanin, PC, and then enters photosystem 1. Photosystem 1 operates very much like photosystem 2 that we've already looked at, whereby a photon of light excites an electron in chlorophyll, and the chlorophyll passes that energy as resonance energy until it reaches the chlorophyll molecule in the reaction center. Again, the reaction is elevated to a primary electron acceptor, and there's another electron transport chain. The second electron transport chain, however, does not produce ATP. Instead, the electron ends up on NADP+, reducing it to NADPH. Now, NADPH is very similar to NADH and FADH2 in that it's an electron carrier and moves high-energy electrons to the Calvin cycle. So just to recap and take a look at this, putting all of the molecular machinery in the context of the thylakoid membrane. Light strikes photosystem 2 and causes an electron to be excited out of the reaction center into the primary electron acceptor. After this happens, water is oxidized to replace the electrons that have left the reaction center. Electrons are then passed to plastoquinone and then to the cytochrome complex, which pumps protons into the thylakoid space. The proton gradient here is used by ATP synthase to generate ATP, and this is produced on the stroma side of the membrane. The electrons continue from the cytochrome complex to plastocyanin and then to photosystem 1, where their energy is elevated again by another photon. As with photosystem 2, the electron is taken up by a primary electron acceptor and passed to ferredoxin and NADP+ reductase, which causes the reduction of NADP+ to NADPH. ATP and NADPH are both produced in the stroma, and they're passed on to the Calvin cycle, where they deliver their energy in order to fix carbon. Now, this flow of electrons through these two photosystems is known as linear electron flow, because because of the way the electrons move in a single direction through these complexes. In some cases, there's an alternate flow of electrons. After the electrons have been elevated through to the primary electron acceptor in photosystem 1, they may be passed back to ferredoxin and back into the cytochrome complex and the electron transport chain. This results in production of ATP, but no production of NADPH. Also, because photosystem 2 is not used, there's no oxidation of water and therefore no oxygen produced. In some organisms, this is the only route that electrons take through photosynthesis. It's thought that this is probably the original form of photosynthesis that photosynthesis took when it first evolved. Organisms that contain both photosystem 1 and photosystem 2 can use a combination of linear and cyclic electron flow and can therefore adjust the ratio of ATP to NADPH produced. So far, we've seen the light reactions and how water is oxidized into oxygen, and how ATP is produced through chemiosmosis and NADPH is produced at the end of the linear electron flow through photosystem 1. ATP and NADPH provide the energy required to fix carbon from carbon dioxide molecules into sugars, and this happens in the Calvin cycle in the stroma of the chloroplast. There are three phases to the Calvin cycle. In the first phase, carbon fixation, carbon dioxide is attached to ribulose bisphosphate, RUBP, using an enzyme called Rubisco. RUBP is a 5-carbon molecule, and with the addition of a carbon dioxide molecule, the resulting molecule has 6 carbon atoms. That 6-carbon molecule is then broken into 3-phosphoglycerate, which only has 3 carbons. The next phase involves the reduction of 3-phosphoglycerate to produce G3P. This reduction is very energetically expensive, and so both ATP and the electron carrier NADPH deliver their energy in order to allow this to happen. In the third and last phase, multiple molecules of G3P are joined together to regenerate RUBP. Again, this is energetically costly, and so ATP is used to fuel the endergonic reaction. Now, you might notice that the number of molecules involved in the Calvin cycle seems kind of weird. In order to balance these equations, we need to enter 3 carbon dioxide molecules and turn the cycle 3 times. This generates 3 6-carbon molecules and therefore 6 molecules of 3-phosphoglycerate. The reduction step results in 6 molecules of G3P. One of these molecules of G3P exits the cycle and is used along with other molecules to form glucose and other molecules. That leaves the cycle with 5 molecules of G3P, or 15 carbon atoms in total. These carbon atoms are rearranged into 3 5-carbon molecules of RUBP. So, in order to output a single 3-carbon molecule of G3P, 3 carbon dioxide molecules must enter the cycle, and the cycle must turn 3 times. So, we've seen how the light reactions and the Calvin cycle work together to produce sugars. Ultimately, light provides the energy in the light reactions to energize ADP into ATP and energize NADP plus into NADPH. These 2 high-energy molecules are used to provide an energy boost that's required in order to fix carbon dioxide and produce reduced high-energy sugar molecules.